Hello, good afternoon, everybody, and welcome to this latest webinar from the Francis Crick Institute. Hopefully you can all see that splash screen there. There's a couple of buttons you can use during this webinar. Please use the chat function if there's any issues with the sound, you want us to speak more slowly or more loudly. And if you've got any questions, please put those into the Q&A box and we'll probably deal with those towards the end of the webinar. So if we move to the next slide. Hi. Right, so today we're sort of carrying on our journey through flow cytometry. Those of you who have been to some of these webinars that we put on over the past 3 or 4 months will be aware that we've started with basic flow cytometry, an introduction to fluorescence and fluorochromes. We've looked at panel design, we've looked at compensation and data analysis. We've looked at some particular applications. But we've also looked at a couple of parallel technologies. So we've seen the rise in recent years of imaging flow cytometry, which we looked at, and mass cytometry. And one of the hotter topics of today is spectral flow cytometry. So actually, I should have introduced myself. I'm very remiss there. My name's Derek Davis. I'm the STP, or Science Technology Platform training lead here at the Francis Crick Institute in London. And what I've done with some of these webinars is actually invite guests in to help me. So today I've invited Shonna Johnston, who works at the University of Edinburgh. And there you can see Shonna.
Hi, Derek.
And Shonna will be actually giving this webinar today. So I'll hand over to Shonna and I'll come back at the end because we have some list of resources for you at the end, and I'll also help handle the Q&A. So all yours, Shonna.
Thank you. Thanks. Hi, everyone. So I was going to say morning, but I guess it'll depend on whether it's afternoon or morning where everybody is. So today, I'd just like to thank Derek for inviting me to speak, and I hope you're all surviving this lockdown world that we're in just now. So I'm going to give a bit of an introduction to spectral flow cytometry and the experiences that we've had in my facility. So because travel is restricted just now, I thought I'd give you a wee introduction to my hometown, which is Edinburgh. The picture here of Edinburgh Castle is one that I took towards the end of last year. And obviously that's what everybody knows about Edinburgh, big castle sitting on the top of a hill in the centre of town. But the University of Edinburgh is obviously quite an old university, and we have the Old College, which is situated in the centre of town. But the campus that I work on is the Little France campus, and I work in the Queen's Medical Research Institute, which is a QMRI, which is why our title is obviously QMRI Flow Cytometry Facility. Now it's called Little France because we also have another castle right beside us, which is Craigmillar Castle, and Mary Queen of Scots stayed there quite a lot, and with her French entourage camping down on the fields beside it, that's why it's known as Little France. So to give you an idea about our labs, we hold a couple of the internal labs within the QMRI, and you can see we're a team of 3. That's myself with Will and Mary there. We're actually part of the Centre for Inflammation Research, but we are obviously available, our equipment and our expertise is available to the whole of the Institute and other areas of the university and some commercial labs as well. So we are mainly a BD lab, but we did expand out a little bit in 2018 to have an Attune and the Aurora spectral cytometer. So I should say now that I've tried to keep it as generalised as possible, but quite a lot of my examples are actually from the Aurora itself. So in recent years, cytometry has expanded quite dramatically. So we've obviously got our conventional cytometry that we all know about, and as Derek says, mass, imaging and spectral. So spectral seems to be, I guess, the one of the sort of more baby of the family. So hopefully I'll give you a decent introduction to today. So my first experience with spectral analysis was actually when I was facilitating with the imaging facility within our institute. And you can see here, this was an article from 2005, and actually the instruments, commercial microscopy instruments doing spectral analysis were available then as well. So that was my first experience using an LSM 510 Meta and a Leica SP5. Now, there was a flow cytometry spectral analysis article came out in 2006, but an actual instrument didn't come out until 2013. So that's quite interesting, the sort of disparity between those two times. And essentially the difference with it is that flow cytometers, they had to have a massive increase in their speed and their sensitivity to be able to actually do spectral analysis well compared to with the microscopy solutions. So we move forward now to today, 2020, and the first spectral analyzer that you saw in the previous one was actually a Sony spectral analyzer. They've now added to their portfolio with a couple of extra ones, most recently the ID 7000. You've got the Cytec Aurora here, and we've also got the Bigfoot by Propel Labs. So as I said earlier, my experience is obviously of the Cytec Aurora. So what is the difference between spectral and conventional flow cytometry? Well, actually, most of it is not that different. So your fluidics, that part of everything is actually quite similar. The bit that's different is actually the detection. And you can see that where conventional flow cytometry uses dichroic mirrors and bandpass filters to select certain ranges within the spectrum, with flow cytometry, what we have is we've got dispersive optics, which can actually send it across a detector array, which allows us to measure the full spectra from all the particles that are coming through the instrument. So an easier way to look at this, I think, is to use like a normal spectrum viewer. And if you have a look, you can see the small area that's picked up here compared to these multiple small areas that are picked up here. Now, one of the things with it is that with spectral flow cytometry, the bandwidths are much narrower. So because those bandwidths are narrower, the amount of light that's actually collected in each one is much lower. And that is why there needed to be a massive increase in the actual processing speed and sensitivity of the instruments to be able to do good spectral resolution of populations with flow cytometry. Now, one of the things that you've got to be aware of with these cytometers is the number of detectors that are actually in the system. So the standard 3-laser Aurora has 38 detectors in it, and it goes up to 64 for a 5-laser system. With the Bigfoot Propel one, you've got your 60 detectors, and the latest Sony is 188 detectors. But the biggest thing that everybody has to be aware of is that the number of detectors does not equal the number of fluorochromes. That was a massive thing that we had to sort of get our users to understand. Although one of the good things about it is obviously you've got more data points. So you should have a more accurate mathematical calculation of the fluorochrome signal, which is good. And possibly because of the extra photons that are light across, that are collected across the spectrum, then you would like to think that we might get some tighter clustering in there as well, which would be great. So how do you prepare your panel for spectral flow cytometry? Well, it's different, but similar to what we normally do at the moment. You've still got to have a good understanding of your actual instrument itself. You need to understand what the lasers that you've got on your instrument are. Filters are obviously not so important. Because it's a spectral flow cytometer, your filters, you should be covering the full spectrum with them. But what you do need to have a good understanding of is these different spectral profiles and how they're produced. The different instruments, the different manufacturers, they have slightly different takes on what the spectral profile is. And actually, you can even see here, the differences between a 3-laser and a 5-laser system are quite significant, I think, as well. So you need to be able to understand it. You need to be able to read it. Is it collinear lasers that you're working with? Are the arrays specific to the lasers or are they not? And as I say, it's really important that you actually have some understanding. You can see here, if we look at the violet laser, it's actually associated with 16 different violet detectors. And the blue is associated with 14 detectors, and your red is associated with 8, and that is what is creating that spectral profile. So it's not that it's down at the bottom end it's 450 and at the top end it's 800. It's right across the spectrum from sort of 410 right up to 7 sort of about 800 here, and then from this one from about 500 up to 800 and so on. And that kind of thing is really important to get a grasp of before you start doing spectral flow cytometry with your instruments. It's also really important, again, as you would normally, it's really important to understand what your fluorochromes are doing. Do you know, what are they excited by? What is their emission? And again, the emission is obviously related to that spectral profile, so it's really good to understand what's going on in there. And you can see we've got an example of FTSE here, and then we've got an example of PE. Now, if you compare the staining indexes of a lot of these fluorochromes compared to your standard systems, there may be some discrepancies in that. And again, you've got to be aware of what lasers you have. Your PE is obviously not going to be as excited as well by a blue laser as it is by a yellow-green laser. Now, the other interesting thing is that actually if you try to measure AF532 on a normal flow cytometer with a blue laser, I can guarantee, having tried it a few times for people, it doesn't work very well at all. And even things like tomato and cherry can be a bit hit or miss, I would say, sometimes when you've got a blue or a yellow-green, with only a blue laser. However, in these systems, even though we only have the blue and we don't have the yellow-green, you can see they're actually measured, no problem at all. They might not be so bright, but you can actually pick them out from a sample. And again, as I say, the staining index might be a little bit different to your conventional cytometer, but what you're actually interested in here is obviously doing the same as you would normally. So you would always choose your a weak antigen, you would combine it to a strong fluorochrome. Or in this case, you might want to choose it, select it with a fluorochrome that's got a unique spectral profile, because that's actually what you're separating on. It's not it is obviously strong and weak is important, but actually unique is important too. And that's to try and make sure that you've not got too many issues with spreading. And again, you would have to think about your co-expression on these as well. You don't want to choose highly overlapping fluorochromes on antigens that are co-expressed. So these are just examples that have come from the SciTech resources. They're quite easy to find online compared to a few of the other companies. They don't have as much resources online that are easily accessible to anybody. And again, spreading, you still definitely get. I do have an example of this a little bit further on. But the nice thing about it is that actually with panel design, there are a few more new tools that the companies have brought out to help us with choosing our panels. Now, the first thing I did was I decided to Google Spectral Similarity Index, and actually it came up with the Oscars webpage, because apparently they use Spectral Similarity Index to describe lighting for digital cinema cameras, which is quite interesting. And actually it's doing exactly the same thing as what we would want it to do in flow cytometry, which is measuring how close a given spectrum is to another spectrum. Okay, in this case, it's a single value that represents the quality of the curve fit. What their single values are, I don't know, I didn't go into that, but I did have a look at the flow cytometry example where the scale goes from 0 to 1, where 0 is no similarity at all to 1 being identical. So if we have a look at these two here, we've got your BV421 and your AF647. There's absolutely no similarity between these two peaks at all. And you can see that represented here within the similarity index. If we go to something like BV421 and super bright 436, you can see the similarity is pretty dramatic and actually is pretty close to 1 on this similarity index. So what does that mean? If we have a look at that again, I think it's quite nice to be able to look at it in a spectrum viewer. I think that makes sense to people to do that. If you were using a conventional system and you had just a 450/50 bandpass filter in place, you wouldn't be able to discriminate between those two fluorochromes at all. However, if you start looking spectrally at them, you can see the difference that you're going to be getting here. So this is actually me taking the SciTech bandpass widths and just uploading them onto the BioLegend spectrum viewer, and you can see all the little band widths coming in here. Now, the interesting thing that I'm just going to point out is that there is no one here. Okay, there's no bandpass that covers this range here, and that's because you're wanting to avoid any kind of backlight coming through from the 488 laser. So that's why there isn't one in there. But you can see there's a definite difference between those two spectrums. And if we have a look on here, how does it look? So this is an example of your BV421 here. You can see it's quite high in V1 and V2. Whereas if you look at super bright 436, you can see a slightly higher peak on the V2 channel. So if we have a look at those two against each other, you can see the spreading is fairly dramatic, which is one of the things that I was saying earlier. You've got to make sure that actually when you're choosing your fluorochromes, that you are still choosing them in a sensible manner and that you're not just choosing things that, you know, might work. It is always quite difficult to do that with researchers, but it is really, really important that you don't just assume that because it's spectral that you don't have any issues with these types of things. The other nice design tool for spectral flow cytometry is a complexity index. So what you can do is you can take two different panels here. And again, in this one, what I've done is I've kept the 421 and the 436 in it, and it's got a complexity index of 7. Whereas this one, which is a little bit better spread out across, is down at 2. The recommendation for this is that a well-designed 9 or 10 colour panel should only have a complexity index of about 2 or 3, whereas a large panel of 35 colours and more, you would maybe expect a complexity index more of about 40 or 50. So that's quite good. But what does that mean for us? Well, if we're looking at a conventional flow cytometer, and you would be looking at a different panel, a panel on here, you can see what you would be wanting to do is spread your fluorochromes within the laser, and you'd be wanting to spread your fluorochromes across the laser. But you can see that we've got a few of these where they've got very similar filters that we're going to pick them up from, even though they're not necessarily excited by the same, mainly picked up by the same laser. So on something like this, you might not think this is a great panel, okay? I mean, it will work, but it's going to need a certain amount of compensation. Obviously, the more you fill up your detectors, the more you're going to end up with higher amounts of compensation. I don't think it can be avoided that much. However, if you compare it to the uniqueness of the fluorochrome spectral profiles, then you can see the complexity index on a spectral flow cytometer would suggest that this is actually a pretty decent panel. So one thing I think that's really important is that you can't just assume a good filter-based panel is going to work on well on a spectral flow cytometer, and vice versa. It's really important that people don't think they can just move straight from one machine to another, but it is definitely worth looking at it and seeing where you're going to go with it. So you're happy with your panel, you've got your antibodies ordered and they're arrived. Where are you going to go next with it? Okay. You need to think about how you're going to do your sample preparation. The one thing with the spectral flow cytometer is it's not just some magic box that you can put just junk into and you're going to get some really nice data out of. You still have to work hard to make your science good, to make sure that you're actually, collecting some decent data. So when it comes to sample preparation, it's still really important to make sure you've got that single cell suspension. Flow cytometry, that's what it's all about. We need our samples to be in a single cell suspension, or otherwise, what kind of data are we going to get out of there? It's important to make sure your yield is right. With some of our groups, we work on liver cells, and we have maybe about 3 or 4 different protocols that are more specific for the different types of cells that we actually have in the liver. So again, that's really important to make sure that you're getting decent yield of the cells of interest. It's incredibly important to make sure that you are preserving your epitopes and that you're not losing them during your protocol. And again, viability, massively important. The nice thing actually with the spectral flow cytometer, I don't know if I've got an example here, but certainly in some of the researcher examples that I've got, we can actually measure APC, AF 700, APC size 7, and we can add in a near infrared viability marker as well, which is nice because it just expands that your capability of the red laser quite a lot compared to what you would have on a conventional flow cytometer. So again, really important to make sure that you get all of those things right. And again, when it comes to sample preparation, what's important? You still need all your controls. A lot of our guys, as I say, they kind of thought, they were coming to me and they were saying, oh, would you really like to use the spectral flow cytometer, Shonna? Does that mean we don't need to bring FMOs and isotypes? Of course not. You still have to make sure that you're going to be bringing all of these. Now, the thing I've got up at the top here with the unstained, I always like to run an unstained anyway. I think it's really, really important to show what your base layer, your baseline is of the cells. And it's a nice reassurance that actually your stain sample has worked properly. Is it more important for spectral flow cytometry? Well, yeah, I think it probably is. I know in a lot of places they don't want you to say that things are more important than spectral compared to before, but in this case, I think it really is. If you're going to extract the autofluorescence from the samples, then it is really, really important that you actually know what the baseline is on that to start with. And actually, you can't extract the autofluorescence unless you have an unstained there. So you still need your single stains. It's difficult. Do you want to go with beads or with cells? For me, we are doing some testing, and I'll show you some data from that later on. But to compare the spectral profiles between beads and cells, but I've just helped somebody run an assay yesterday where they used cells as a single stain on a conventional flow cytometer. And actually, it was really, really difficult to set the compensation on that because of the autofluorescence of the cells. Are you going to be trying to take a stained F480 macrophage and then align it to a lymphocyte? That's impossible to really be able to try and figure out. So that's why my preference in a lot of cases is to work with beads, because the autofluorescence of the positives and negatives are obviously the same, and you always get a definite positive and you get a definite negative in this case. And my feelings on this and how we do our sample prep does impact how we actually do our spectral flow cytometry as well. And again, I'll go into that a little bit further on. Controls, obviously you still need FMOs and isotypes, and absolutely you need a stain sample. So how does spectral analysis affect your actual workflow on the systems? Well, if you're working with a normal filter-based system, the first thing that you actually have to do is run a full stain sample, and you need to be able to set those voltages. Some people obviously run a CST, you can do filtration assays, that type of thing to be able to set your voltages. But if you put a sample on there and things go off scale, you have to be able to bring them on, or otherwise your data doesn't mean an awful lot. So it is really important. I mean, obviously there's some cases you can get away with it, but in general, we would always have to make sure that you run a full stain sample to be able to check that those voltages are going to work for you. Okay. You would then go to run your unstained. As I say, I like to make sure that actually the sample is properly stained, and then you would go to run your single stains with automatic compensation. Now, as I say, your compensation is directly dependent on your voltages, so you cannot go to that next stage until you've run your got your voltages right. So you run your sample, you've run your unstained, you're doing your single stains. I personally prefer beads when it comes to automatic compensation, and then afterwards we usually add in the fluorescent proteins and viability dyes if we can into the matrix, and sometimes we just have keep them out of the matrix to start with. We then duplicate those single stains so that we can actually view them in an n by n plot once the compensation's been applied, so that we can actually check that basically verify the compensation is right and it's doing something that's sensible and that it's just not just gone out of whack for some reason. So then you would go on to your sample run. So if we have a wee look at the n by n plots, this is what we would normally do. So we would take it as it is without compensation on it, then we'd apply the compensation and see how good it looks. So because your compensations are directly dependent on your voltages, you can't assess a panel until after your stain sample has been run. So it doesn't matter how much theory you've gone through. I mean, I've seen some panels that in theory look absolutely dreadful, but work really, really well. And some panels that you think are going to work really, really well that don't quite live up to that expectation in practice. But when it comes to spectral flow cytometry, where are you going to go with your workflow? So the first thing that I really like about it is that we don't have to worry about any kind of configurational changes. We don't have to change filters or anything like that in there. And I always have this, I like to mention something about a shape sorter block, because I am always quite amazed at how many people can't put the filters back in the way they should, even though we have pictures in the machines to show them how to do it. But the nice thing with spectral flow cytometry is that you're measuring everything in it. You're unmixing it against references that you obviously have in there. So if you've forgotten to choose a channel because somebody has told you it's a 10 colour panel and actually it's an 11 colour panel, you don't have any issues with this. You can even edit that experiment post-acquisition if you want to, which is great. But the main thing that you've got that differs between spectral flow cytometry and your filter-based systems is with the adjustment of the gains and voltages. It's so important that you preserve your spectral profile. So what I did is I just did a little test on the machine, and I was looking at GFP beads. So if we have a look at the GFP spectra, so this is a reference spectra that I looked at just from the Cytec website, so I had some idea about what I wanted it or what I should expect it to look like. I then ran some beads, and you can see we've got quite a nice GFP signal here. So that's great. If you want to start adjusting voltages, how do you go about doing it? Well, one of the things that I did was I just started playing around, and obviously with the Cytec it's an APD, so it's gains that I was playing around with, but you can see I was playing around with the gains, and actually I can completely change that spectral profile. Although in a normal filter-based system, it's really important that you get the optimum voltages and that everything is working nicely according to your cells as well. If you start really changing the gains and everything around on these APDs, you will dramatically change that spectral profile. So it's not a good idea to do something like that. What we want to do is we actually just want to make adjustments, maybe by a certain percentage across all the detectors associated with a specific laser. So in this case, what I've done is I've reduced everything of the blue laser only by 50%. And you can see it's still preserving my spectral profile. And that's really important for something like this. So essentially, when you go to run a spectral flow cytometer, you're not going to be automatically going in and changing voltages and APDs as much as you might have wanted to do on a filter-based system. So what does that mean for us? Well, it means that actually we can run beads, and we can set up reference spectras for experiments and check how well a panel is going to work together without even running any kinds of samples at all. That has made quite a dramatic difference to our users, because rather than having the stress of actually trying to set up samples for collection, as well as then setting up, seeing if their panels worked and things like that on the machine, we can actually run through a set of beads, put them into the reference, and then what we do is we create an experiment that has those beads again. So we rerun it as an experiment, and then we look to see what that algorithm is doing and whether it's unmixing it well. I've got a little bit more about algorithms a little bit further on. So these types of things are really important. What we're doing is we're actually running the reference spectra, we're checking them against the known spectral profiles for those particular fluorochromes. And then what we can do is we can create a bead experiment only and unmix it using the library. That gives us a little bit of confidence in the actual algorithm itself and how well it's working. It gives us confidence in the library. And then we can use single colour multiplots to actually separate it out to see how it's looking. So that's quite nice. Then only after that we can then run the samples. Okay, so then we can run single stain cells if we want to. We can run our controls and we can run the actual sample and see how we get on. You don't, as I say, we use the library quite a lot because, and we do use beads quite a lot. I work in a department that's quite heavily interested in macrophages and things, and we'll work with tissues quite a lot. We don't get an awful lot of cells back. I mean, I totally appreciate if you're working with human blood samples and you're looking at lymphocytes, monocytes and granulocytes, that you probably have quite a lot of cells that you can work with and you can easily separate out your populations. That's great. If you can do that, brilliant. But for us, it just isn't quite part of our normal workflow. So if we have a look here, you can see our single channel multiplots, and that's just allowing us to say that, yeah, that what we would expect to see with the compensation actually works with an unmixing. So how does all that actually work in the background? Well, if you look at compensation, the main thing to remember with compensation is that you've got two data points and you're essentially moving them into alignment. There was a paper recently that's just come out saying about how they're looking at it across a continuum, which is quite interesting, and they're setting it up and they're saying that actually compensation hasn't moved on in 30 years in flow cytometry, which obviously it hasn't, and then they're trying to look at this different way of doing it, which is quite interesting. How we do it and what the way we show people to do it is that we actually do those end by end plots and we leave it so that it's uncompensated to start with. Okay, by duplicating our beads all the time, we're allowing people to actually see what the automatic compensation or the spectral unmixing, how it's actually working and how is it affecting the samples that we're running through. So if we take this sample here, these are APC beads that I've run through around through the instrument, and I was looking at them just against a few different fluorochromes, other channels. And you can see you're almost getting a very, very basic spectra across here. And then when you add on the compensation, you can see you should only see populations coming in here and nothing coming in these other areas. Now this is great for our users. That actually really helped us move from filter-based systems onto spectral analysis, because they had some understanding about what things are doing. And actually this is pretty much what how it works with the Bigfoot, and maybe even if you're trying to do spectral flow cytometry using, say, a symphony or something like that, you're looking at the non-primary detectors for the fluorochrome that you're interested in. Now, the nice thing with compensation though is that at the end of it, it gives us a number. So we've got all these different kinds of matrices that we get from the different instruments or in the different software packages, and we understand what these mean. We're used to seeing them, and a number gives people a certain amount of confidence. However, with a spillover matrix, you don't get that. Everything is just zero, okay? And that, I have to say at the beginning, was incredibly disconcerting for us, because you're relying on the algorithms for the unmixing, which actually, because of the workflow that we have in place for our filter-based systems, it wasn't too difficult to then move to the spectral analysis workflow. So what is happening? With compensation, we've got this set up here. Now I quite like this diagram that I got from the Sony website about it, but you can see that bandpass filters in a normal filter-based system, your bandpass filters are collecting emitted light at defined sections along the spectrum, with each filter is related to a detector which is specified for a fluorochrome. And out with these areas, there isn't any information acquired at all. So you can see that in detector 1, say, it's mainly just the green fluorescence that we're seeing. And then you can see in detector 2, it's mainly the yellow with just a little bit of red and green coming through. And in the last detector here, detector 3, it's mainly red that we're actually seeing. However, with spectral flow cytometry, you've got to know your spectral signatures. So you've got all of these in place here. And what you're doing is it's actually summing it up. So while it sums it up, you can see you're getting this overall spectral capacity coming in here. And then how do you decide what is coming from which fluorochrome? That's why you need your good reference spectras in there. So there's 2 different kinds of unmixing algorithms, and as I said in the talk the other day, I am really not a mathematician, but I had to look into it a little bit more, so I had a little bit more confidence into what I was actually understanding from it. Now, with an ordinary least squares algorithm, it's an unbiased method that assumes normal error distribution across the wavelengths, whereas a weighted least squares allows for unequal measurement error. Now, the Cytec uses the ordinary least squares, and the Sony prefers the weighted ones. Why is that? I think what's actually important is what detectors are in the system. So the difference with the 2 systems is that with the Cytec system, you have APDs, whereas with the Sony system, you have PMTs. So the APDs have definitely got improved red sensitivity, although we have to appreciate the dark noise of the APDs. But if you have a look across here, you've got your APD is coming across this direction here, and it's quite high all the way across between your 400 and your 800 nanometers. Whereas with the PMT, it drops quite rapidly down to the 800. So what happens is that when you're doing your unmixing, you can assume almost equal weighting across the APDs because of this almost straight line across this part here. Whereas with the PMTs, you have to weight it because it's actually dropping down towards the 800. So how much impact the different spectra has depends on what the wavelength is. Grand. So when it comes to data analysis, where you're going to go with it, we actually do most of our unmixing on the cytometer workstations, but both FCS Express and FlowJo have got some kind of unmixing capacity within them. FCS Express have obviously been working quite heavily with Cytec to the point where you can actually import raw Cytec files, and they are also using the ordinary least squares method. So it means that their algorithms are fairly comparable, and the data that you get out at the end is fairly comparable. With FCS Express, you can also generate a cross-stain index and a spillover spreading and matrix for your Cytec Aurora too. With FlowJo, there's what's called a spectral compensation, and it features in versions 10.6 and above. And essentially what it's doing is it's utilising signals from the non-primary detectors, as I sort of alluded to earlier. And it's just a slightly different way of working it on there. As I say, our experience is mainly off FCS Express for spectral flow cytometry compared to FlowJo. So when it does come to your unmixed data, there are a few things that it's really important to get right. So one of the things up here is that actually you can have multiple types of unmixing. Generally, you would have maybe one where you've extracted the autofluorescence and one where you've not extracted the autofluorescence. But then maybe you've used one kind of reference spectra or one set of reference spectra for one unmixing, and then decided that actually you wanted to try a different set of reference spectra for another one. Maybe you've been using the library, but actually you've decided that you don't want to use the library, it's not quite appropriate in this case, and that you'd rather just use a brand new set from your experiment. It's really important to make sure that these are all well labelled. You can see here, unmixed 1, 2, 3 and 4 is not going to get you anywhere when you're trying to figure out where you're going with your data at this point. It's also really important to understand that actually your unmixed files are much, much smaller than your raw data files. Your raw data files have got as many data points as you have detectors, whereas your unmixed data files have only got the number of points that are actually according to the number of fluorochromes that you have in the panel. So they are quite a bit smaller. Here's a couple of examples that I've put in here. But the thing with the unmixed FCS files is that actually it can be analysed by any standard flow cytometry analysis software. It's not going to make it weird because it was spectral in the first place, because everything does just turn into dot plots as we would normally understand. So what are the practical benefits of actually having a spectral flow cytometer? Well, there's a couple of nice little experiments tests that we did on to start with that I'll show you, which is verifying spectral profiles of commercial reagents. And then I'm going to go into the autofluorescence extraction quite heavily, because that is something that was quite huge for us, made quite a big difference. I've got a couple of larger panels that I can show you from some of our users, and then obviously there's a bit of in-house testing that we're doing as well. So if we have a look here, this was actually one of our users who brought in a brand new PE size 7 antibody, and we were getting really, really weird results coming up on our normal filter-based system. So what we did was we said, okay, we'll have a wee look at it on the Aurora. So that's what we did. So you can see we've got this spectral profile coming through. And what we did was we compared it to what we think a PE size 7, a known PE size 7 should be. Okay, so we took this from the Cytec resources, and you could see there was a massive increase in this bit here, which is probably coming from PE. And my guess is that actually the antibody had degraded quite heavily already. So they were able to send that back to the company with the information that we got from here, and we got a new antibody, which was great and was way improved on the first one. Something else that we did with it is we quite often use GFP beads. So we have commercially bought ones that we use for doing compensation on our filter-based systems. They work grand. What you're looking for is the difference between a negative and a positive. Or is it, in the filter-based systems, actually what you're looking at is not between a negative and a positive, it's between a lower one, a weaker signal and a higher signal, just 2 data points. That's what's actually more important that you're looking at for compensation. So when we're normally picking out the P2, we should be our negative beads, and the P7, it works absolutely grand on filter-based systems. However, when you're doing spectral flow cytometry, if you want to do some kind of unmixing, so extract the autofluorescence from this and set it up, you can't, because actually the dimmest one, which should be just a background, it should just be autofluorescence of those cells and not have a GFP spectral profile, doesn't. It actually does have a spectral profile. So you can see, if we look at these are just P2, 3, 4, 5, 6, and 7, according to these ones here. And you can see it looks exactly the same as the rest, it's just weaker. So that was a bit disappointing because that gave us a bit of problems when we were trying to look at GFP, especially if you've got very small amounts and it's quite difficult to define in a population. So autofluorescence, that is our big thing that we're really interested in. Because we work with macrophages so much, you can see that if you've got a bright signal, great. If you've got a dim signal, still okay if you don't have high autofluorescence. But if you've got high autofluorescence, then actually you're going to impact pretty severely on any dim signals. I actually did a test with some monocyte-derived macrophages, and 6 out of our 18 channels on our 5-laser LSR were essentially affected by autofluorescence interference. So 6 out of those 18 channels, you had to have a pretty bright fluorochrome on them, which is a shame because actually some of those are pretty decent channels. The fluorochromes that go into those channels are usually quite decent ones. So yeah, it wasn't great for us. It essentially took our 18 colour instrument straight down to a 12 colour instrument, which wasn't so good. If you have a look at it on the spectral profile, you can see how high they look. So to go into autofluorescence extraction in a little bit more detail, as I say, my first sort of foray into this was with imaging. And you can see, if you look at a normal wide field system, you can actually see the difference between what is autofluorescence and what is GFP signal in here. Whereas if you go to confocal, the confocal is just using a PMT, so it's just picking up photons, okay, and the colour is just an add-on afterwards. So it can actually define between those slight differences in spectra that the wide fields will pick up. So that's not so great. So what do you do about it? With confocal, what you can do is you can do exactly the same as what we do with flow cytometry. So you can do the spectral analysis of it. And if you take a pixel here, then you can look at the autofluorescence spectral profile. And if you take a pixel here, then you can look at the actual FITC fluorescence that you've maybe got, and you can see that it's localized to the nucleus. Now, it is really, really important that they are spatially separated, and it's also really, really important to know that actually you can only do this autofluorescence extraction if it is true autofluorescence. You can't extract it if it's non-specific binding, or if you're fluorescent antibody, or if it's secondary staining. Okay, it does actually have to have a different spectra. So if you do autofluorescence extraction by confocal, this is what you get. So it looks lovely. And actually the reason I did this was because somebody wanted a last minute experiment for a paper, as you can imagine. And what they wanted to do was they had kidney and they'd stained up their macrophages with FITC. So the kidney autofluorescence was way outweighing the actual signal they were getting from the F480, and we were able to change it around a little bit so it looked more like that. So it was quite good. Flow cytometry wise, what does that mean? If you look at some just unlabeled and per-CP labeled calibrite beads, this is the unstained ones, and these are the per-CP ones. And if we have a look at the the spectral profile of these, the unlabeled has the spectral profile. The per-CP, you can barely distinguish it. It's a tiny little sort of area maybe that we're getting up here. And if we run these both together, the unlabeled and per-CP beads together, you can see they're not a massive difference going on between them. So if you just run them normally, that's what it looks like. Whereas if you actually do autofluorescence extraction, you can massively improve your signal to noise. So that's actually given us an improved stain index just by itself, just by taking out the autofluorescent extraction. So what I'm going to do next, that's obviously quite a big part of our stuff, is I'm going to give you some idea about the research that we've been doing in our department. So apologies because this is quite a busy slide, but it was given to me by one of our researchers. And essentially what they were looking at was normal and low density neutrophils. The gating strategy isn't really that exciting, to be honest with you. They were mainly looking at differences in activation markers between the two. And then they were also looking to see if there was any good combinations of markers that would help them if they ever wanted to go to sorting. And lastly, what they also did was they used the autofluorescence extraction or the autofluorescence measurements to plot between the normal density and the low density neutrophils. They didn't actually see anything, but I was quite interested to be able to have a look at that on there. For this next one, we're looking at mouse liver myeloid cell panels. And you can see we were getting really nice setups in this. When we first did the demo of the Aurora, the guys basically took what was in the back of the fridge and set up panels from that. And we found that actually some of the clustering they were getting was improved compared to some of the filter-based systems that we were working with. Now, Prakash, who gave me this data, he actually said it is, although he didn't include it in the slide that he gave me, he did say that it was worth highlighting that the AF channel, the autofluorescence channel, did help him separate the myeloid and the lymphocytes, the lymphoid cells much better, and that although again he's not shown it here, that he was able to separate the lineage positive compartment quite easily into T cells, B cells, etc. This last panel here is quite a nice 20 colour panel that Diana from our institute was working with. She actually gave a talk at Cyto last year, and it was all about validating our single cell RNA sequencing results using spectral flow cytometry. So one of the things I am going to mention in case anybody notices is she did use BV650 as our dump, but what she was doing as our antibodies were biotinylated and she was coming in with a streptavidin 650. So there shouldn't have been too many differences in that tandem dye that she was using there. And then she was able to look at this sort of setup in here. So what she was doing is she was using the Aurora to validate single cell RNA sequencing to identify macrophage subpopulations in the liver after paracetamol-induced liver injuries. So you can see here that she was able to define all the different cells that she was actually interested in and then compare it to her RNA sequencing data. So in-house testing, we've obviously been doing a little bit to see how well the machine's working so that we can test it and give people advice on what we think it's useful for. So we were looking at reference spectras, we've been looking at unmixing, autofluorescence and viability dyes quite a lot as well. So one thing with the reference spectras is we were interested in differences in the spectral profile when the antibody is bound to a bead or a cell. As I said, I quite like using the beads for normal filter-based systems, but we were a little bit concerned that that might be something that would have an impact. I haven't seen a massive amount of difference with quite a few of the fluorochromes that I've been working with, but I haven't done a huge test. A lot of these things have been put paid to by the lockdown. We were just getting into quite a nice program of testing and then it all sort of got stopped short. For reference spectra, we're again interested, is there a massive difference in spectral profiles between unfixed and fixed beads and cells? Should you make sure that actually if you're fixing your cells, you fix your beads as well? We think possibly that is true, but again, we just like to make sure that we're actually proving that to ourselves as well, which is important. It's also really, really important that we're going to check these across time as well to see how reliable our reference library actually is and where we can go with that. But again, it is quite important to actually just run bead experiments again and again to make sure that that reference library is working if that's what you choose to go for. So in this example here, I was lucky enough to get some beads from Simon in Australia, who was able to, so I could look at tomato and pea. We had one of our groups was really interested in tomato positive cells from their line that they were looking at, their mouse line they were looking at, and we were wanting to see if we could measure it against pea because they also wanted to measure an antigen that only worked on a pea antibody. Unfortunately, it doesn't seem to work with any other kinds of fluorochromes, and actually you can see you can distinguish those really nicely, which is quite good. Autofluorescence, as I say, was a massive thing for us. So for our monocyte-derived macrophages, we were interested in whether the treatments actually change the spectral profile of the macrophages, but actually it doesn't. On the different treatments that we tried, we looked at the spectral profiles of quite a few different ones. And the examples that I've got here are with dexamethasone and with interferon gamma, LPS treated ones. And actually we found it was a difference in intensity as opposed to the actual spectral profile that we were seeing. But again, this is data we didn't have before, so it is quite important to know that it is still data, even if it's not making any changes. For viability dyes, one of the things I'd been starting to work on with Roy Edward from Biostatus is we were looking at just what kind of concentrations we should be using. We actually looked to see that if we use JAK5 at the recommended concentration that you would maybe use for a filter-based system, and it did stay on scale in a filter-based system, and maybe we should put that on here. It was actually going massively off scale. So those types of things are quite important. And we also realised that carryover was something we had to be aware of as well. So we had to do a few sit flushes in here to make sure that that worked. These are all just our little bits of testing just to see how we could advise our users. The other thing with viability dyes is we were also interested in what autofluorescence is with cell death. Obviously, it's one of the things I've always said for years is that autofluorescence increases as your cell starts to die. So are we able to distinguish that in any way, and can we use viability dyes to actually pick that out a little bit better? So again, hopefully I'll have a bit more data and by the end of this year, if I can get back into the lab a little bit more on these types of things. But essentially, although spectral flow cytometry hasn't actually redefined the science of flow cytometry, what it has done is it's given us the ability to measure and analyse information that we didn't have previously. And in addition to increasing panel sizes with fewer lasers, I'd like to think that all it's going to do is increase our knowledge of the cell and its functions, and obviously that's what we want to try and do as well. Okay, thanks very much.
Thank you, Shonna. Just before we've got a few questions that we can ask you. Just before we go to those, just to, as I mentioned, a few other resources for you. So as well as following the Quick Training Twitter account, you could also follow the RMS or Flow Cytometry UK. If you want to see what's coming up in terms of training at the Crick, if you go to the What's On page, everything should be there. And if we don't manage to answer your questions now, if you think of anything a little bit later on, I'm sure both Shonna and myself are quite happy to answer any questions by email at a later date. And what I will do in the next couple of days as well is send everybody a link to the recording of this video, and also a link to all the other videos or webinars that we've done over the past couple of months. So let me ask you a couple of questions, Shonna, while we've got you here. So the problem we had when we introduced imaging flow cytometry, for example, is everybody suddenly wanted to use that.
Yes.
What would you say to somebody who'd got a reasonably well-designed panel and they're running it on a conventional cytometer that suddenly says, I want to use the Aurora, I want to use a spectral flow?
So yeah, I will say yes, absolutely. I think it works really, really well. I think you just have to be careful. If you've got something that's already working on a filter-based system, it obviously depends where you are in your projects. We have quite a few people who are quite far on in their projects and don't want to move across to a spectral system because the data was going to be quite different for them. So obviously they wanted to continue on with using the LSR, which was absolutely fine. And then the biggest drawback that we actually have is that we don't have a matched sorter. I think that will make such a massive difference to people if they'll be able to do some matched sorting. So have an instrument that's actually matched to the Cytec Aurora that we can actually do some sorting with. But aside from that, it's really easy. I have to say, I think spectral flow cytometry makes a lot of sense. It does help that actually the software is pretty intuitive. And although in some regards you don't know what you can get away with, so one of the things that people are a bit worried about, I think, with spectral flow cytometry is that actually they think that they're going to have to be more specific, or they're going to have better controls, they're going to have to bring better samples and stuff like that. They should have good samples in the first place anyway, obviously, but you don't necessarily need better samples. It just needs to be done well in the first place. And the other thing is, if you don't know what you can get away with, we're all used to filter-based systems and we know what we can get away with on those, whereas you don't know what we can get away with on spectral flow cytometry. It's too soon, it's too too new to all of us to actually decide what we can get away with. And I mean, that is what people do try and do, obviously.
Yeah, exactly. I mean, I don't know whether you're aware of any studies or done anything like this, but it'd be nice to see a direct comparison with sort of a conventional cytometry system that has APDs like the Cytoflex and something like the Fortessa, because that spreading error is all down to photon counting error, or mainly down to photon counting error, and that's going to be different. So it'd be nice to see the comparison.
Yeah, so one of the things that we, as when we were doing the in-house testing, is we actually started buying in quite a lot of antibodies that were just CD4, so that we could actually just do bead tests between them and see how we were getting on with it. And then we were trying to obviously compare them with lymphocytes to see how that was going. I know that actually there's quite a few companies that are beginning to sell multiple lots of CD4 conjugated to all different fluorochromes now, which I'm guessing is for a lot of these reasons, so that you can actually do better comparisons between your instruments. As we all know, even if you've got two LSRs that are set up configurationally the same, there'll be some differences between them as well. And I guess that's the kind of thing that we're I know quite a few flow cytometry managers are actually trying to figure out where the systems fit within their or where they sit within the labs, to see what the comparisons are. And I think that it's the same with spectral flow cytometry and filter-based systems. I think you still need to have some idea about where they actually sit for you.
And I suppose we have to get used to maybe slightly different rules, because compensation is not the same as spectral unmixing, is it? So
no, it is quite different. And I think that was the thing that we found quite off-putting to start with, because when you have a number and you know what that means, and you can increase and decrease that number, even though you shouldn't, if you are increasing and decreasing it, you can see what's actually happening with it. Whereas if you're just given a 0 and you don't know what's actually got you to that place in the first place, it does make it quite difficult to define how it's worked. And I'm a firm believer in that people shouldn't just assume things are a box and take the numbers out at the end of it. You actually have to have some understanding about where that data is coming from and how you got to that point, because I think it is really, really important that you actually get the best out of all these instruments.
Okay. And one sort of, again, maybe a final question of specificity to the tandem dyes, because you showed an example of a tandem dye that had oxidized, or you're getting a lot of the donor left. Does that mean you have to, same with compensation control, you have to bring the same tandem dye that you're going to use in your experiment for your reference?
Yeah, so actually the nice thing with it is that say you have multiple ones, you can actually create, because you've got your library, where you're going to put all your reference spectra, you could even have 2 lots of the same antibody if you wanted to, so that you could actually compare the differences in that. And I think that's going to probably, I don't think you'll see the differences in lots quite so much in a filter-based system as what you will on a spectral system. I think you'll actually be able to define the spectral profiles of all your antibodies much, much better. And I have been interested in doing that. It's just finding a whole load of PE7 antibodies and seeing if I can run them through and what they actually look like.
Yeah, once you get back into the lab, there's your project, right?
Yeah, that's it.
Okay, so I think we've come to the end of the questioning. So as we said earlier, if you've got any questions you think about later on, please feel free to email Shonna and myself. But thank you all for coming. We hope to see you again at a webinar in the very near future. So thanks everybody. Stay safe.
Cheers, thanks. Bye.